So thanks to the organizers. As a Tulane alumnus, it's great to be here. Always feel right at home. I also represent Auburn University as Associate Dean for Research for the College of Sciences and Mathematics. But today I'm representing Terra Bioforge. I'm going to tell you about engineering bugs to make more drugs. So the story starts in Wisconsin. As a postdoc, I worked on metagenomics, ways of cloning DNA from diverse microbes to make natural products. I had a very good fortune to meet Dr. David Mead, who was the founder of Lucigen. And together, we worked on this kinds of technology. David sold Lucigen for more than $70 million. 2016, we started the company called Verizymes. For your nucleic acid amplification and diagnostic needs, go to verizymes.com. So Verizymes, its greatest product so far was an enzyme used for COVID-19 detection methods. And so that went so well that we spun off the drug part of Verizymes into Terra Bioforge in 2021, together with Nancy Keller and Neil Kelleher, who are natural product chemists and fungal metabolite experts. So the drug side of Terra Bioforge is what we'll talk about today. The value proposition here, there is an over $1 trillion market for natural products. The market for microbial secondary metabolites, small molecules, is about $250 billion. This technology addresses pharmaceuticals, agriculture, animal health, consumer needs. And if you think about the issues here, why are prescription drugs so expensive? And why is there a drug shortage? The technology Terra Bioforge has developed enables us to mass produce drugs that are more expensive to produce through chemical synthesis. Our technology involves cloning biosynthetic gene clusters. These often can be 150 kb, 150,000 base pairs. These megasyntheses are very complex. We use a CRISPR-Cas9 system in order to clone the biosynthetic gene clusters, or BGCs, and then into patented expression vectors that allow us to then move the DNA encoding these drugs into different expression systems, including both bacterial and fungal expression systems. And so you can see Bacillus, Aspergillus, Streptomyces, and gram-negative bacteria can serve as the expression host to mass produce these compounds. We engineer those strains to make even more compound. For example, our patent-pending microdrive technology allows us to mass produce particular compounds using a fungal host. The idea here is that microdrive allows us to integrate that BGC multiple times within the genome of the fungus, especially in transcriptionally active sites. So microdrive, you can see here the bicoverdin, you can see the red pigmented compound being produced, and the bicoverdin is an antibiotic and anti-cancer compound. And you can see visually that this microdrive technology helps us to mass produce this compound using this fungal expression system. If you compare the production of bicoverdin using single native or single integration, we get about 35 times more product compared to other competing methods. There's only one other company in the world that has this kind of technology, that's Ginkgo Bioworks, and this technology is unique to Terra Bioforge. So we enable, through these big DNA technology tools, to mass produce drugs more rapidly, and then it's very scalable. You can go from small scale fermentation to large scale fermentation. Here are our existing clients. A lot of what we've done to develop the company is contract research. We've produced over 150 different natural products from a variety of companies and academic institutions. They retain all IP. This is a good deal for both of us. This has helped us to develop the company and be able to validate our technology through contract research. For example, vancomycin is one of the compounds we produced. These very complex small molecules that are very expensive to produce, we can produce much less expensively. We have extensive intellectual property, but I would say our most valuable resources are trade secrets. This is a very difficult technology to do from start to finish. Terra Bioforge is not going to put in a patent those trade secrets that would allow our competitors to compete with us. Here is a pipeline of our billion-dollar compounds. As we've been working on natural product production for other companies, we've also been developing our own company assets. You can see the SBIR supported, these are phase 1 SBIR supported grants from NIH. We also have SBIRs that are submitted. And you can see each one of those compounds are company assets with very extensive markets. And these are compounds that we're advancing with company resources. Ultimately, this is where we're going, is using the contract research to keep the lights on while we develop our own company assets for these particular anti-cancer, immunotherapy, antifungal agents. The idea here is to develop compounds that have already been de-risked. We can produce these compounds at a fraction of the cost of chemical synthesis. This is a great value proposition. So here's where we are in terms of our company development. We've had a $3 million angel round that has allowed us to do technology validation, and the custom service, the contract research, has enabled us to validate this technology and to de-risk this technology. Now we have a list of assets that we're using SBIR funding to help develop, and we're looking for funding in the about $5 to $10 million. The $3 million angel round was an evaluation of $6 million. We're looking for $5 to $10 million of investment at a valuation of $15 million. And so the idea is to focus on commercialization of these compounds that have big upsides. If you look at our revenue projections in millions of dollars, our contract research revenues, we think we can grow organically over time. That business is growing about twofold every year. They have a lot of contracts just coming in from India. But really, it's the licensing revenues from our company assets that are really going to grow the company. And that's where we see the big upscale in terms of eventual acquisition. The idea here is to have a exit upwards of $300 million based on comparable companies that have been acquired in recent memory. So we feel like we're in a very good position given that we have a validated technology. We have a lot of contract research for production of natural products. I think we're a very attractive target for investment. Here's our team. As I mentioned, Dr. David Mead, he is really the brain, the guru of this, right? So David was the inventor of TA cloning, which has generated more than $1 billion for Thermo Fisher. And this is the fourth company that he's co-founded. Linda also has taken a life sciences company through exit. And we have an amazing team of PhD scientists in Middleton, Wisconsin, as well as a great board of directors and scientific advisory board. So here's the big picture. We have a very experienced team, CEO and CFO that have started life science startups and exited those. This is a pretty low development risk. We've already validated the technology. We have many different natural products that are being produced as company resources. And if you think about pharmaceutical industry pain points, we can reduce the cost of goods dramatically, right? In terms of the cost to produce these natural products. This is a big need for us in terms of providing more cost-effective pharmaceuticals. And we offer currently that as a service, right? For many different life science companies, pharma, big and small. And so this is also a pretty mature technology. This is something that's very difficult to copy. I've been working in this field since 1998, and it's really exciting to see where we are today. And I'd like to thank Patrick and the organizers for giving me a chance to talk to you today about Terra Bioforge, and I look forward to your questions. Thank you.
Yeah, we've got about a minute or so for questions. Anyone?
Thanks for the talk. I was curious for the molecules that you're developing in-house, what's the, so like manufacturing, there's always a lot of CapEx, know-how, scale-up difficulties, purification and all of that. What's your guys' plan for that? Are you guys going to be developing that internally, trying to fund that out, or like getting CROs or?
Yeah, partnership CRO side. That could be a partnership where really large fermentation capacities are being used for something else, right? They can be used for mass production products. Think about the fungal expression of our best technology. That's very compatible with yeast fermentation. So the other nice thing about producing these compounds in scale, when we sometimes the wild type strain will produce many different derivatives. whereas the expression in the other systems, you see less privatization. So that might be useful in clinical trials. We also think that can help us in terms of scale of production. So, time's up.
Thanks.
Thank you.